Today, I will be speaking about the impact of patient engagement and patient centricity in clinical development of gene therapies. I am Oscar Segurado. I'm the Chief Medical Officer of ASC Therapeutics. This is a biotech organization based in the San Francisco Bay Area, focused on programs for gene therapy, gene editing using CRISPR-Cas9, and also allogeneic cell therapies. Today, I'm going to be discussing with you one of the most important aspects on clinical trials, and this is the most relevant element to make sure that a clinical trial is successful. And this is everything related to patient centricity and the new concept of decentralized clinical trials, or DCTs. I will discuss a meta-study of over 4,000 clinical trials, and I will discuss with you key elements and perspectives. I will give you as an example the gene therapy for hemophilia A that we are developing at ASC Therapeutics, and I will make sure that you see a number of conclusions and next steps that are relevant from the clinical development perspective. Starting with patient centricity and decentralized clinical trials. The background of this information that I'm going to be discussing with you is the fact that most clinical trials struggle to enroll patients on time and on budget. The average pivotal trial collects around 1 million data points, and 1 out of 5 trials are terminated due to under-recruitment. Once the trials begin, an average of 30% of the patients are going to drop out. In a meta-study from the Economist Intelligence Unit, it was analyzed the results of about 4,000 clinical trials with a number of patient-centered elements, and it was compared with 20,000 traditional clinical trials that were conducted between 2012 and 2017. The outcome of this study was that the patient-centered clinical trials had 87% of positive results, compared with 20% less, about 70% of patients that really contributed to positive results. The implications of this type of analysis is that there is a growing emphasis on patient engagement and patient centricity, which is at the end of the day one of the most promising and exciting new direction in clinical development. So what are the key elements of patient centricity? The most important aspect is that we need to ensure that the patients are empowered. What does it mean? It means that in today's environment, patients are very, very savvy. They already are able to understand their disease. They understand all aspects of clinical trials, and we need to recognize and leverage this patient expertise. Recognize that when they come to us, when we ask them to engage with us and consent to participate in a clinical trial, these patients have already been speaking with colleagues, with family, with their physician, so that we are talking in some way to experts, especially for patients that have been living with their disease for many years. We also need to ensure that when we incorporate the patient in our clinical trial, we are also empowering their social network, the way they interact with their family, friends, with other patients, and definitely with the environment from a healthcare perspective. So we need to engage all these other elements or aspects that are relevant for that patient. And definitely it's very important that we consider the importance of patient advocacy organizations. And I'm going to give you some examples on our area, which is, again, everything related to hemophilia A. So we need to promote true partnerships with these organizations to ensure that there is a kind of feedback loop, and patients and these advocacy organizations are working together. So from the perspective of decentralized clinical trials, although at the end of the day, they are also going to be relevant to the patient, what that means is that it's not only the reference center where usually we perform or we conduct our clinical trial that is going to be relevant. Also, we need to recognize and leverage the expertise that is already there in this therapeutic area. We need to understand that nowadays, with all these digital tools, wearables, the information that practically everyone has with them with a smartphone, that we are able also to provide the patient all the tools that are necessary to ensure that this particular trial is not only taking place and being related to the principal investigator, that potentially the patient could even receive some type of follow-up at home or with other satellite sites. So this, therefore, is important to consider both empowering the reference site and the satellite clinical centers. We need to engage the entire network, including the PIs at the reference center and at potentially satellite sites. We need to empower healthcare providers and payers that also support the study sites, obviously working as sponsors with the clinical research organization, the CRO, and ensure that there is a continuous flow of information, that there is a continuous learning, especially on everything related to digital and mobile approaches. So let me give you some key perspectives for clinical trials. First of all, we need to ensure that we educate the patients. The patients are savvy, as I mentioned a moment ago, but we need to leverage these patient advocacy groups. I'm going to talk about the National Hemophilia Foundation in the United States. We need to also consider even day-long dress rehearsals to explain these patients how the trial will work. We also need to educate the clinical sites. We need to focus obviously on the principal investigator and the study team. We definitely have already explained everything in terms of ensuring that the entire network around the patient is well aware about the consequences, about these study visits that need to take place. And definitely we need to spend some time on these IT, information technology tools that we are having and we're using. This is what I call high touch and high tech. We need to make sure that the patient also is able to sign this electronic informed consent, that they understand what a patient-reported outcome means, maybe using wearables, maybe using an e-diary, and also ensure that all these outcome assessments are very clear to the patients, so that there is also a homogeneity around patients when we are collecting all this data. Absolutely relevant here is that we educate clinical trial sponsors and definitely the CRO. So we need to accompany, to engage all these stakeholders in a kind of cultural transformation on what nowadays we talk about patient centricity and the centralized clinical trials. The remote monitoring protocols should be very clear also to the traveling nurses, which nowadays are becoming more and more involved in these clinical trials. And definitely we need to ensure that everything that we're doing also reflects what we call a real world data capture. So it's not only all the tests or all the aspects that are related to monitoring the patient, also to have everything related to real world evidence. Now, let me spend one moment about, or a section of this presentation on gene therapy for hemophilia A. hemophilia A is considered a rare disease because it's one of approximately 7,000 different rare diseases that have been already identified, and we know that they exist today. If we consider all the patients that are affected by these rare diseases, and by the way, about 30% of them will not be able, in many cases, these patients to live until even the fifth birthday, we will see that about less than 10% of all these rare diseases have an approved treatment available. So the FDA has approved over 600 orphan drugs since the passage of the orphan drug. But what this means is that when we take into account all these rare diseases, we're going to see that this is a therapeutic area on itself, because about 10%, 1 in 10 patients in the US and in general in the rest of the world have any type of rare disease. So what is hemophilia A? Hemophilia A is a disease where there is not enough or not enough elements, what is called the coagulation cascade, so that the proteins that are required for the clotting of the blood is not taking place, so that the patient eventually could go into bleeding or uncontrolled bleeding, which happens in internal organs and in the joints. So although factor VIII is synthesized in the liver, is not always synthesized in the hepatocyte. So there are two types of hemophilia, hemophilia A and hemophilia B. Hemophilia A is the one I'm going to be focusing on, and this is when there is a factor VIII that is missing. And also in hemophilia B, it would be factor IX. One characteristic of all these patients is because they have this type of serious clotting defect, they need to have a replacement therapy. That means that they receive either factor VIII for hemophilia A or factor IX for hemophilia B. And the majority of these patients develop inhibitory antibodies. And all these patients that have already inhibitory antibodies are going to be in trouble when we provide them any kind of replacement therapy, especially when we do gene therapy. So what this means is that we have to take into account all that, and many of these patients will not be able to receive these type of therapies because of these inhibitory antibodies. So worldwide, about 400,000 patients have hemophilia, and the majority of these patients are men. This is an X-linked chromosomal defect. Now, what do we do with gene therapy? I like to use as an analogy the Apollo 11 journey to the moon, where you have the capsule and we have the lunar module. And that's what you see here to the left. In some way, the capsule is in the case of gene therapy, a viral capsid, which encodes and has integrated all the information of factor VIII. And this is what we would call the lunar module. And to the right, you see that this capsid is incorporated into the liver cell. It goes into the endosome, is delivered to the nucleus, and in the nucleus, it just delivers the DNA, and the DNA is not incorporated into the chromosome. It's called episomal, but is able to produce the protein that is going to be making in some way factor VIII from this hepatocyte. There is another alternative, which we're also delivering in our organization, which is when this capsule would be landing in the moon. That's the way I describe that. And this is when we are able to take this DNA for factor VIII, and we, in this case, incorporate that into the chromosome. This is very important because what we do with that is that the corrective factor VIII DNA or transgene is stably integrated into the genome, and this is important for pediatric patients. Why is that? Because pediatric patients have a liver that is growing, and as such, the cells divide. When the cells divide, if we use a standard gene therapy, this information from the DNA is lost. So that's why this program that we have using CRISPR-Cas9 is able to provide the so-called cut and paste. We look for a specific site in the chromosome, which is close to the albumin locus, and we use the promoter of this albumin so that we're able to start producing factor VIII indefinitely in the individuals. We are doing this work right now only in animal models, but the gene therapy is well advanced, and we are already in our clinical trial for the phase 1 and phase 2 trial. In the case of this CRISPR gene editing, the advantage again is the integration, and this is why we need more time, because we need to confirm that this type of approach is safe and effective. So for all this information, we need to make sure that the patient is well educated. We need to explain that these patients require genetic testing, that we need to make sure that we understand what are the specific defects that these patients have. We need also to explain them the importance to identify if these patients have pre-existing antibodies to the gene therapy product. And we need to ensure that these patients have the right level of severity so that these patients are going to be able to take this therapy in relation to other therapies that are already being delivered to these patients. So very important for these patients is to ensure that they understand that we are already working with them in terms of safety. That's why these clinical trials have a very, very thorough monitoring plan. In our case, for instance, we look specifically into imaging methods, into biomarkers, into ways to identify how the liver is performing, and also the possibility, very unlikely possibility, of this liver developing any type of neoplasia. We also have, like in other clinical trials, a very strict and rigorous evaluation of safety over time. This is pharmacovigilance, and we are required by the FDA, although our endpoint is about 1 year, we are required by the FDA to follow up for 5 years, and after that, even the majority of these patients are going to go into registries. So in some way, patients on gene therapy are going to be followed up for life. So this is all the information that we have in terms of safety, but we need to explain them what are the advantages that they're having. Again, we're talking here about patient centricity. I'm giving you a sense about how important it is to explain to the patients what they're going to get when they get these kind of therapies, and how important it is to explain to the patients all aspects related to the primary efficacy endpoints. What are we expect to see over usually the first year, what we expect to see in terms of follow-up over a number of years. And very important nowadays in gene therapy is durability. And this is what we are bringing in our case, and you're going to see in a moment the way we explain that to patients with a second generation gene therapy, which we have already seen that requires lower dose of these patients, also lower cost, and a longer durability of the therapy. We as sponsors need to identify what aspects of our therapy are going to be relevant to the patients, and definitely the patients need to be clear that we need to collect all information in terms of longitudinal profile. So the way we do that, and I'm going to give you an example, is that we provide these patients videos and printed materials. At the top, you see here a 4-page brochure that we have already developed, where we explain the concept of hemophilia A, we talk about the challenge, the solution, and we provide all the information that they need to ensure that they understand what is needed. Also, we have created a video, which I'm going to show you right now.
Did you know that over 400,000 people worldwide live with hemophilia every day? People with hemophilia lack factors essential for the coagulation or clotting of blood, which can result in serious bleeding in joints and other internal organs. Hemophilia A is the most common form of hemophilia. This rare genetic disorder is caused by defects in a gene that is essential for the production of a coagulation protein called factor VIII. This genetic defect exclusively affects the X chromosome, therefore mostly affecting males. Current therapies replacing the missing coagulation factors are not a long-term fix, and in fact, people with hemophilia must receive these treatments during their entire lives, requiring regular therapeutic interventions, often burdensome, painful, and expensive. But now, novel gene therapies are changing the way we treat hemophilia. First generation gene therapies for hemophilia have been investigated in the past few years on hundreds of patients. One of these therapies for hemophilia A is under review by the U.S. Food and Drug Administration and approved for clinical use in the European Union. A one and done infusion incorporates the corrective gene into the patient's liver, which becomes a biofactory that produces the missing coagulation factor, potentially for life. ASC Therapeutics has developed a second generation gene therapy for hemophilia A that, in extensive preclinical studies, showed a significant increase in the secretion of factor VIII from the liver into the bloodstream, therefore requiring a low dose and potentially increasing the durability of factor production. This investigational gene therapy has been cleared exclusively for clinical trials in the US by the FDA Food and Drug Administration. Once the corrective gene is transported into the liver cell, the process of producing functional factor VIII protein takes place in multiple compartments of the cell. Importantly, the normal factor VIII protein needs to be released into the bloodstream for it to carry out its coagulation function. Maintaining a healthy lifestyle is critical to ensure that people with hemophilia can both produce and secrete factor VIII from the liver biofactory in the long run. Gene therapy is a promising therapy that brings new hope to people with hemophilia A. Patients, together with their families and doctors, will potentially be able to live a normal life without all the burdens of current hemophilia therapies. Speak to your doctor about the clinical trial and contact ASC Therapeutics for more information today.
So as you can see, it's extremely important that we use lay language, that we explain to the patients what they need. So in conclusion, I would say that sites and patients must understand the impact of the clinical trial. They need to know the results of individual biomarkers, genetic tests, understand the individual side effects, which study arm they're enrolled, if they're a placebo or a comparator arm. They need to know that the clinical trial results are going to be presented in the public domain, obviously not their actual individual data. And companies and sponsors must have a seat on the table for patients to share their voice. We need to represent them and to have clear that there should be a two-way dialogue. Patients must be able to express their day-to-day unmet needs. We're providing information, but we want a dialogue. We want to get also information from them. And definitely patients will benefit from a convergence of clinical care and clinical results. So in general, in terms of next steps, I think that the clinical trial sponsors and monitors should take a holistic perspective of patients and as individuals and trial participants. We need to think about them all the time. We need to ensure that sites and patients consider themselves as an integral element crucial to the success of the trial and advances in medicine. Patient advocates and organizations should have an independent voice with regulators, including revised endpoints. So what that means is that if we see over the conduction of the study that we need more information and we need to change aspects of the clinical trial, we need to do that always considering the voice and the feedback from the patients. With that, I would like to thank you so much for your attention.